Today I want to defend my proposal research, which is entitled Bioremediation of Trinacan Lactate Wastewater using Endophytic Fusarium Species. So what is lactate wastewater? Lactate wastewater have a strong relationship with accumulation of solid waste. The reason why I propose this research because I have observed that there are great accumulation of lactate wastewater in Trinacan, Sarawak. In addition, lactate wastewater have unpleasant smell and can cause water pollution. So I think lactate wastewater need to be managed efficiently and wisely. Besides, I need to consider the cost and the technique must environmental friendly. So I propose to do bioremediation by using endophytic fusarium species. My objective for my research, I want to analyze and assess this very potential bioremediation agent for lactate wastewater treatment. Secondly, I want to identify the enzyme that contribute and involve in bioremediation action on lactate wastewater treatment. And lastly, I want to observe and identify the best condition for fusarium species to done the bioremediation process effectively. I have 4 subtopics in literature review, which is lactate wastewater, bioremediation, endophytic fusarium species, and lastly, past research on bioremediation by fusarium species. Firstly, lactate wastewater. Lactate wastewater is produced when there are percolation of water in a collection of solid waste. This is happen when the waste is undergone decomposition by the microbial agent. Lactate wastewater become a serious issue in Malaysia because it can cause water contamination and water pollution. It also have a very bad odor and strong color. Lactate wastewater contain many physical and chemical contaminants. Based on my reading in several articles, they reported that there are 4 main contaminants in lactate wastewater. Firstly is heavy metal. Second, unrecyclable plastic and materials. Third, lactate wastewater have very high reading of COD, BOD, and pH. Lastly, lactate wastewater have almost to 20% of agricultural waste. So all of these components in lactate wastewater can be harmful towards all the living things, especially humans and animals. In fact, all of these contaminants also can cause the treatment of lactate wastewater become complicated and difficult. So we go to bioremediation subtopic. Bioremediation can be defined as a green alternative process to degrade toxic and very high-risk environmental contaminants by naturally or intentionally occurring of microorganisms in order to degrade and break down the substance to a safer and harmless product. It can be included by using bioaugmentation, bioabsorption, and bioaccumulation. In early bioremediation application, they are using aerobic and anaerobic microorganisms for the waste treatment. So there are several advantages of bioremediation, which are bioremediation is highly effective and efficient. They are more economical, which is cost saving. The technique is more environmental friendly and widely approved by the community. Several articles mention that there are 2 major techniques of bioremediation, which is ex situ and in situ. Ex situ can be defined as my bioremediation process above the ground, which is we done the treatment in another place, not the polluted place. In general, ex situ bioremediation not use any microactivity in waste treatment, while in situ bioremediation is fundamentally underground process, which is we conduct the treatment at the place itself. And it is vice versa with ex situ bioremediation, which is an in situ bioremediation use the microbial activities to enhance the waste treatment. So I propose that my research is under the in situ bioremediation because I will use and I will study the fusarium species on liquid wastewater treatment. However, bioremediation itself have its own limitation. First is bioremediation process have longer period of treatment time. The second, the process is so sensitive towards the environment condition, which are temperature, pH, toxicity level, as we use the microorganism, right? So different microorganisms have different optimum activities. And the last one is generally bioremediation process is hard to control and monitor. So we go to the next subtopic, which is endophytic fusarium species. So fusarium species is a filamentous ascomycete and from Sordaromycete class in order Hypocreals and from Natriaceae family. Endophytic fusarium species is different with general and common fusarium species because endophytic fusarium species only can be found in healthy plants and usually isolated from the plant's root. Many research reported that fusarium species is a very potential and useful as biological agent in many industry, like textile industry, pulp and paper industry, food and beverage industry, and many others. In addition, fusarium species have contribution in biofuel, in medical and biotechnology field. This is because they contain abundance of bioproducts such as enzyme, bioactive compound, bioflavors, pigment, and biofuel. Besides, fusarium species can be a good resource of extracellular enzyme, as some research mentioned that it can break down a complex polysaccharide, lipase, and protein, as they have a capability to produce enzyme such as lipase, amylase, beta-glucosidase, beta-manase, nitrilase, and many others. This indicates that it's a good biological agent for bioremediation on liquid wastewater in Ternate, Sarawak. So I will proceed on the next subtopic, which is past research on bioremediation by fusarium species. Firstly, the research that undergone by Asra and Sachireka as 2016, it mentioned that an endophytic fusarium species, which is Fusarium oxysporum, is a potential fungus to degrade heavy metal. Secondly, based on Roma et al. Research in 2019, hydrocarbon treatment, specifically microremediation of naphthalene, was successfully done by Fusarium oxysporum. L. Said et al. 2020 has been improved that Fusarium solani successfully can biotransformation and bioabsorption of zinc. In addition, Fusarium species have sustained ability and potential to grow and reproduce in media with all of those contaminants. Fusarium oxysporum has been used for cyanide contamination treatment in Ziames, which is corn. So we proceed to material and method. For site description, I will undergo this study and research in molecular genetic laboratory in Faculty of Resource Science and Technology. And then the leachate sampling, I will collect leachate wastewater from 3 different locations in Trinidad. For the microorganism, specifically the Fusarium species, I will obtain the Fusarium species from the molecular genetic laboratory fungal collection. And the fungus, the Fusarium species, will maintain on mold extra agar as I have sampling it, and I will undergo the characterization of the leachate. I will filter the leachate samples by using the Whiteman 0.5mm filter paper. After the filter, I will conduct the analysis by referring the examination of water and wastewater ABHA method, standard method. After that, I will study the tolerance of leachate wastewater, where I will dissolve different concentration of leachate samples in medium extract agar. I will also prepare control media to study the tolerance. So I will put the specific Fusarium species at the center of the leachate base agar. To observe it, I will measure the radius of Fusarium species mycelia. I will measure it every 48 hours for 2 weeks. I will proceed to bioremediation screening with free biomass. I will place the inoculum culture of Fusarium species in 20 ml of collected, untreated, analyzed leachate wastewater that I collect from sanitary landfill, secure landfill, and wastewater treatment plant. Each type of leachate wastewater, I will prepare 3 replicates. So the biomass are incubated in various temperature, with at 10 degrees Celsius, 30 degrees Celsius, and 60 degrees Celsius. So for screening on different pH, we will repeat the method yearly, and we use different pH, which are pH 5, 7, and 10. To observe the result, I will analyze the enzyme and color change daily, while COD and toxicity of the leachate base agar I will evaluate it at the end of experiment. So after I conduct the bioremediation screening with free biomass, I will undergo enzyme assay to observe the presence of leachate enzyme. I will identify by using colorimetry. Because leachate can decolorize synthetic dyes like azo, malachite green, and Congo red. The decolorization is indicate the oxidation of aromatic and phenolic compound, while peroxidase enzyme will be observed by the color change using spectrophotometric method. And lastly, scanning electron microscopy. The inserted and non-inserted biomass of endophytic Fusarium species with leachate, I will prepare with 6% of glutarialdehyde. I will incubate this biomass at 4 degrees Celsius for overnight, and I will wash it with phosphate buffer until 2 to 3 times. After I wash it with phosphate buffer, I will dry the biomass by using critical point drying. I will coat the biological sample. After that, the sample will be observed by using the scanning electron microscopy. For the expected outcome, I expect that the isolated Fusarium species can be a potential and successful bioremediation agent for Trinidad leachate wastewater treatment. Besides, the important physiochemical bioremediation parameters can be identified and optimized. For my work schedule for final year project, I will done it from September 2020 until May 2021.